Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel Diversity of Plants Zone. In this channel you can easily find the videos related to plants and their diversity throughout the world. Today's topic of my video is about evidences of palynology, genetics, reproductive biology and biogeography in relation to the taxonomy. So let's start today's video. First of all, I will describe about the pollen factor. Palynology is the science of pollen and spores and its applications. It is derived from the Greek word palynion, meaning to gather, to spread and to distribute, in recognition of the fact that many pollen grains and spores are easily carried by wind. The significance of pollen attributes in taxonomy has been realized during the last 3 decades. Now I will discuss about the number of the nuclei in the pollen. The number of the nuclei in the pollen at the time of dispersal has been used by the taxonomists like Brubaker in 1967. The angiosperm pollen is either the binucleate or trinucleate according to the precocity of division of the generative nucleus. The binucleate condition is considered as more primitive than the trinucleate. In centrospermy, the pollen is uniformly trinucleate. The monocot, Liliaceae, is binucleate. The apetalous and polypetalous dicot are binucleate, and gamopetalous members are trinucleate. Now the question is, what is meant by apetalous? Apetalous means having no petals. Polypetalous means having and consisting of separate petals, and gamopetalous means having the corolla composed of united petals. The morning glory is gamopetalous. The shape and symmetry of a pollen grain, the architecture of its wall, exine stratification, sculpture and structure, and type, number, position, shape and structure of its aperture are some of the basic characters which prove useful at all the taxonomic levels. Palynological characters have been used in solving the several taxonomic problems, including repositioning of several disputed taxa and interpretation of problems related to the origin and evolution of the different groups. Cronquist, 1981, and several other workers have made exclusive use of pollen characters in providing classification of angiosperms. Ehrtman used the pollen characters in discussing and solving the taxonomic problems of 105 families. Haywood, 1967, has gone up to the extent of stating that exine details of pollen are such that they can be used in plant identifications, much in the way that fingerprints are used for the identification of criminals. Next I am going to discuss about MONOSULCATE and TRICOLPATE pollen. Jones and Letzinger, 1987, mentioned that angiosperms contain the two basic kinds of pollen grains. First is monosulcate and second is tricolpate. First of all, I will define about the monosulcate. Monosulcate pollen grains are characteristic of primitive dicotyledons, several monocotyledon, tridosperm, and saccharoids. Such pollen grains are boat-shaped in outline and possesses the one long germinal furrow and germinal aperture. The pollen grain is the male gametophytic phase of the flowering plants. Its morphology is highly diversified, from the texture and structure of its wall to its ornamentation and germinal shape. When there is only one polar furrow, the pollen is called monosulcate. Second, I am going to define tricolpate. Tricolpate pollen grains are characteristic of advanced dicotyledons. Such pollen grains have 3 germinal apertures and are lobose in shape. Tricolpate is a pollen grain having the 3 grooves or colpi on each grain, as shown in the figure. This is monocolpate pollen grain because it has only one germinal furrow. and this is the TRICOLPATE pollen grain because it has 3 terminal furrows as shown in the figure. Next, I'm going to discuss about the stenopallineous and unipallineous. Such a taxon in which the type of the pollen is constant and characteristic is called stenopallineous or unipallineous. This type of taxa is homogeneous and a slight variation of the pollen can occur. Example is Gunraceae. This taxon is considered as a very natural taxa. In many types of the plant, the characteristic is always constant. On the other hand, the taxa in which the pollen types vary in size, aperture, exine, stratification, etc., with the grains of various aperture forms, is called the unipallineous. This type of the plant taxa is heterogeneous. In this type of the taxa, the pollen size may vary in the size, aperture, and the forms. The example of this type of taxa is Acanthaceae. NPC-SYSTEM. The classification of pollen is based on the number, position, character analysis called the NPC-SYSTEM. Palynological studies suggest that the taxa with the same journal NPC formula be grouped together and those showing different NPCs separately. NPC-SYSTEM helps in providing a 3-dimensional classification and also in the preparation of the diagnostic case below the family level. Here are some examples of role of the palynology in taxonomy. Author investigated the pollen morphology of 16 species of cypress and prepared a key to differentiate all of them on the basis of the pollen characters. On the basis of apertorial morphoforms of pollen, according to the Mayer, gymnosperms contain alveolar or granular actixine and the laminated intexine, whereas angiosperms contain the columnar or granular actixine and non-laminated intexine. A massive exine and thin intine is present in angiosperm pollens. Several angiospermic taxa have distinct pollen types. The exine pattern is useful in recognizing the different species of genus. Phylogenetic relationships are determined using the pollen characters in several cases. Pollen grains are associated in tetrads in several families of dicots and monocots. Next, I am going to discuss about genetics in relation to the taxonomy. The science of genes, heredity, and variation in living organisms. Molecular systematics, the analysis of the hereditary molecular differences, mainly in DNA sequences, to gain information on organisms' evolutionary relationships. Every living organism contains DNA, RNA, and proteins. In general, closely related organisms, distantly related, usually show a pattern of dissimilarity. Sometimes the DNA assumes the content rate of the mutation and shows the probable evolution of various organisms. The most common approach is the comparison of homologous sequence alignment techniques to identify the similarity. Another application is DNA barcoding, whereas the species of an individual organism is identified using the small sections of mitochondrial DNA or chloroplast DNA. Next is reproductive biology in relation to the taxonomy. Biological systematics is the study of diversification of living forms, both past and present, and the relationship among living things through time. John Lindley provided an early definition of systematics in 1830, although he wrote of systematic botany rather than using the term systematics. In 1970, Machner et al. Defined systematic biology and taxonomy in relationship to one another as follows. It provides scientific names for organisms, describes them, preserves collections of them, provides classifications for organisms key for their identification and data on their distributions, investigates their evolutionary histories, consider their environmental adaptations. The morphology of flowers play an important role in assessing the relationships between reproductive biology and plant systematics. The structure of nectaries in flowers and the composition of nectars provide useful comparative data. Flower fragrances have been shown to influence the behavior of the flower vegetation patterns of the pollinators and can play a significant role in the plant evolution and speciation. Patterns of the UV, ultraviolet reflectance and absorbance from reproductive structures, especially petals, phenological data, pollination and breeding systems are useful for taxonomic purposes. Reproductive biology data and taxonomy provide a better understanding of the function of structural characters used by the taxonomies. It helps to reveal the proper homologies of character states. Next is biogeography in relation to the taxonomy. Biogeography is the study of the distribution of species and ecosystems in geographic space and through geological time. Organisms and biological communities often vary in a regular fashion along geographic gradients of latitude, elevation, isolation, and habitat area. Phytogeography is the branch of the biogeography that studies the distribution of plants. In order to conserve biodiversity, it is essential to know what species are present in a country, province, or area, and exactly where they occur. It is also crucial to know the name of the species because this allows to access to information about whether the species threatened, rare, or of special cultural or biological significance. They not only identify and describe the new species that they discover, but also go on expeditions to survey areas that we know little about. Many taxonomists are also biogeographers. They plot the distribution of different species, identify patterns of distribution, and processes that determine these distributions. For example, temperature and altitude and vegetation. Using the GIS. What is meant by GIS? GIS is a Geographic Information System mapping. A Geographic Information System is a conceptualized framework that provides the ability to capture and to analyze the spatial and geographical data and identify the areas of special importance for conservation. If you like my video, please like and subscribe to my channel. Thanks for watching. Thank you so much.